and then I'll explain what my work is, which is how we achieve precise gene editing in clammy. So Chlamydomonas reinhardtii, which I'm sure you guys are at least a little bit familiar with, because Alfredo and I, like he said, we met in grad school, we were lab mates, even office mates for a while. So this is the organism that we worked with. It is a unicellular green alga, but it is still a eukaryote. Okay, don't assume just because unicellular means it's likely like a prokaryotic, like bacteria or something. Naturally, it is an autotroph, which means it can use light energy to produce and carbon dioxide to produce sugars, basically. But it is also a facultative heterotroph, which means if needed, it can use like an externally supplied carbon source like acetate, which means it can grow in the dark. It's not completely dependent on light energy in order to be able to survive and produce sugars for survival. It can grow on both liquid and solid media, which means, solid media means like on an agar plate, as you can see here in this picture, or in liquid media. And its doubling time is not too bad, although it's not as great as E. coli, which I think has like 90 minutes or something is E. coli's doubling time, maybe even lower than that, actually. So it's about 7 to 8 hours it takes to double. And naturally, they are haploid, but you can also generate diploids. And so all three genomes are sequenced: nuclear, mitochondrial, and plastid genomes. So, trying to manipulate these genomes is actually possible since we have the sequences, and, annotations have improved over the years. And they are transformable. I have never attempted plastid or mitochondrial genome editing. I don't know, Alfredo, did you? I think you tried, you did some plastid, a chloroplast
Yes, I did
transformation.
some chloroplast transformation earlier in my PhD career. Yeah.
Yeah, so I've not attempted that, but anyway, so we do have established protocols for transforming clammy, and targeted genome modifications have been achieved with RNAi, zinc finger nucleases, detail activation. Detail activation was something that one of our seniors in the lab, she graduated a few years ahead of us. She has a couple of papers, interesting papers on detail activation in clammy. And of course, CRISPR-Cas9, which is all the rage, right? I mean, I guess it's been like 10 years since it became a huge thing. But yes, everyone wants to use CRISPR. So what exactly is CRISPR? So CRISPR, those letters in CRISPR stand for clustered, regularly interspaced, short palindromic repeats. Now, what do those words mean? Let's think of the last three letters first, SPR. Think of this as, inside, you're looking at the inside of a bacterial cell that has CRISPR machinery, so all of this. And so before we start, you'll kind of figure this out by the end, but what is this CRISPR machinery used for naturally in bacteria? They use it as basically like a defense mechanism. It's like part of their immune system. It's a way for them to fight off when, for example, a bacteriophage tries to inject its own DNA into the cell and hijack it. They use this method to basically nullify that at the outset. Because once the bacteriophage injects its DNA in there, it basically will take over all the cell's machinery to make more copies of itself. And then, all those copies escape and the cell is left for dead. So don't think of the bacteriophage right now, but think about this, the CRISPR locus. So short palindromic repeats. What is a palindrome? A palindrome is a word or a set of words that either direction when you read it, it's the same. The letters are arranged so that it's the exact same thing. So the word rotator, you can see whether you read it left to right or right to left, it comes out to the word rotator. Same thing is step not on pets. These two I found on the internet, but another common one, the word madam is also a palindrome, right? There's lots of interesting phrases if you Google them. So in this CRISPR locus, these black diamonds that you see are the repeats. They are all fairly short. So I think it's supposed to be like 20 to 40 base pairs, and they are all in the form of palindromes. Why that is important, you'll understand as I explain the rest of the slide. But okay, so that's what the SPR part of CRISPR is. They're short, they're palindromic sequences, and they are all repeats. So all of these repeats are also clustered, right? They are all clustered together. They are regularly interspaced, right? You can see between each of these black diamonds, there are these colored squares. So they are interspaced by what are called, surprise, surprise, spacer sequences. So that's what it is. Your reference point is those short palindromic sequences, which are the black diamonds, and they are all regularly interspaced and they have spacers in between. I'll get to what those spacers actually mean. Now, that's not all the CRISPR locus is. You also have these other genes in here that are called Cas genes. And Cas actually means CRISPR-associated genes. Because you can imagine if this is your DNA sequence that's a CRISPR locus or part of the CRISPR locus, what's the use of simply transcribing the DNA and getting mRNA? You still need to be able to process it. And there are other things that need to happen, right? Because as I'll explain in the next step, these are used as guides in order to be able to cut DNA, but the cutting action cannot happen by these mRNA sequences that get transcribed. You need another enzyme, which is a nuclease, in order to be able to do that cutting work. So, sorry, I lost my train of thought. So anyway, so you've transcribed this CRISPR locus down here. So what do you have now? You have this what is called a hairpin structure or like a stem loop structure that comes out. And you can imagine if this is a palindromic sequence, once it actually gets transcribed, because it's palindromic, you can end up with that hairpin structure where one part of the mRNA is able to hybridize with the other part. That's why you end up with this part in this that's transcribed from the black diamond sequences in there. And what is in between are each of this DNA that is in the squares, the red, purple, orange, green. I'll get to what they mean in a minute. Anyway, this entire sequence is called a pre-CRRNA or pre-CRISPR RNA. So once that is processed, what happens to each of these is they get clipped off basically. So you end up with like this hairpin structure with the tail. Now the hairpin part, can be the same because they're all just repeats, whereas all the tail part of it is going to be unique. And it's unique. Oh, sorry. It's unique. Why? Because the colored squiggly line that we have here will actually recognize the sequence that a bacteriophage can come and inject its own DNA into the cell, so that the color part will actually hybridize with that bacteriophage. So at that point, the mRNA has done its job. It's basically it's found the part of the phage DNA that it can recognize, and it's hybridized to it. Now you see what these oval shaped I don't know, I guess like molecules that are depicted here that are along with the mRNA. Those are the Cas9 that you say in CRISPR-Cas9. So Cas9 is, it'll be one of these genes here. Like there are different Cas proteins. One of them is Cas9. And Cas9 is basically a nuclease. So it's like a scissors. So it forms a complex with one of these CRISPR RNA molecules. The CRISPR RNA molecules will basically drag this whole complex to the phage DNA. It'll recognize it and it'll kind of like latch onto it. At this point, it's brought along with it this scissors, which is the Cas9, and Cas9 will then go ahead and just chop off the DNA. So before the phage DNA is able to come into the cell, get incorporated, it'll go stick itself in different parts of the genome, and then it'll basically hijack everything, it'll make copies of itself, and then it's done its work, and then the bacterial cell no longer exists, it basically dies. Before all that can happen, it basically has stopped all that process by protecting itself against the phage DNA. Now, obviously, to start with, it's not like the bacterial cells will have these, these colored squares, those parts of the DNA already, which is why, there is also like one or probably more than one Cas gene that encodes for Cas proteins that will help in catching bacteriophage DNA as soon as they come in. They will chop up that DNA, but they'll also take it and they'll commit it to memory, basically. They'll take that DNA and they will stick it in the genome somewhere in between these repeats, so that the next time, if a similar bacteriophage with, similar DNA tries to come and do this, it's able to quickly express the mRNA from its CRISPR locus and, bind to it. It'll also express Cas9, which will be one of these genes. And yeah, and that's it. It's able to chop up that foreign DNA at this point. So this is how naturally in bacteria, the whole CRISPR system is basically part of its immune system. This is how it protects itself from foreign DNA coming in and basically taking over the entire cell. I don't think I went over this, but these Cas genes, there are different types. So I know one type of Cas protein is a helicase. So I'm not sure if you guys are familiar, but helicase is basically like an unzipper. So we have DNA is double-stranded, right? Two strands that are hybridized to each other. A helicase will come in and basically help unwind DNA. Yeah, another of course, Cas9 is the main one in this whole system itself, because it will, but because it's the nucleus, so it will cut foreign DNA, while this mRNA piece will guide the Cas9 to that foreign DNA. That's why that's important. Okay, so this is kind of a quick overall picture of CRISPR. Also, I took this picture from Jennifer Doudna's lab's website, and I'm sure you guys have heard by this point, but Jennifer Doudna and Emmanuel Charpentier were their two scientists. Doudna is from Berkeley, and I'm not sure which university, but Emmanuel Charpentier is a French scientist. Both of them got the Nobel Prize for adapting the CRISPR system to be able to use it outside of bacteria, basically. Or actually not, I mean, I think they still showed how you can use it adapted to prokaryotic cells, but to what we want, instead of just the system being able to recognize bacteriophage DNA. So basically what they thought was, you can create, or sorry, you can design that RNA piece that helps guide Cas9 to chop up foreign DNA. It will help. I'm sorry, just 1 second. My computer is complaining about something. All right. Yeah, so they figured you should be able to design part of that RNA so that it recognizes whatever DNA sequence you want it to recognize. And then you can also have this other piece of DNA, which is called the tracer RNA. That's important for the Cas9 to be able to bind to this entire complex. So, sorry, I think I'm getting ahead of myself a little bit here. So let me just go back to the previous slide. So if you remember here, these sequences were called CRISPR RNA, right? The CRRNA. That's actually, there's two parts to that, where we're only depicting one part in that previous slide. So you have this portion, the green portion here is the CRISPR RNA. This brown, or I don't know, I guess like a dark orange portion, is called the tracer RNA. Those two can be designed so that they hybridize to each other. The green portion we already know is of course important because it's going to recognize your target DNA. This brown portion is important because this other part of the stem loop here is where this huge Cas9 enzyme will come and bind. It basically uses that as a handle to form this complex. And of course, the third important part here is this pink blob that you see here. That's the Cas9, which is the nuclease. So the CRISPR RNA and tracer, this is what we call it, the tracer RNA is called, let's just for simplicity's sake, people just call it a guide RNA or a single guide RNA. That's what sgRNA is here. And that guide RNA with the Cas9 enzyme or protein forms what is called a ribonucleoprotein complex because or sorry, so an RNP complex basically. So another important thing to note here is that there is what is called a PAM sequence or a protospacer adjacent motif. You don't really have to worry about exactly what that means. I mean, once you think about it after working with this for a while, it'll kind of make sense. But for different Cas proteins, there are different PAM sequences. So for Cas9, for example, the PAM sequence is NGG. So why is the PAM sequence important? It is important because that's one of the requirements, because it's not just, with in your guide RNA, if this green part is able to recognize your target, that's not enough for Cas9. This is a requirement. You need an NGG sequence here, which is on the strand that is not the one that the guide RNA is binding to. So you see how here this green is binding to this strand of the DNA. On the other strand, you need an NGG sequence where N can be anything. Now, this is actually pretty nice in Chlamydomonas for us because chlammy is a GC-rich organism. So I actually don't remember the percentage, but some large percentage, maybe like 70-something% of chammy's genome is made up of Gs and Cs. So it can work to our advantage, but I always joke that, after I say that, the next time I'm trying to find a good target site, I've basically jinxed myself that I won't be able to find a nice NGG PAM sequence near where I want to target my gene.
Yeah.
So anyways,
So
so
before,
just,
so yeah, so this
sorry.
is, the picture that you showed here, this is the synthetic CRISPR, so something that they modified, right? Not representing the original CRISPR-Cas system that you explained before in the bacteria. So I mean
Yeah, so I think, yeah. So I think in bacteria, it's not necessary that you have like, there's one, I guess, sort of stem loop structure here, and there's one here. I think I'm not 100% sure. I can't remember, but in bacteria, maybe they don't need two stem loop structures. Whereas, yeah, the synthetic system, which, I mean, I hate to say it's only Jennifer Doudna and Emmanuel Charpentier because they built their work on like decades of CRISPR research. But yeah, they were the main ones first who were recognized that they adapted this system where, you can design a piece of DNA that's your tracer RNA that has nothing to do with your target. The important part of this brown part is that it can act as a handle for Cas9 to come and latch on so that it forms a complex. But the green part is important because it has, you have to have part of the green RNA that can recognize your target.
So the green one is the one that's important to guide the edits to be more specific, right?
Yeah. So once this, sorry, go ahead.
And NGG is actually specific for the Cas9, something
Yes.
like recognized by the protein, right?
Yeah. So Cas9 needs it. That is important. And it has to be on the strand that is not the strand that your guide RNA is recognizing.
Oh, okay.
For example, Cas12 is a different type of Cas nuclease protein. The recognition site for that is TTTV, where I think V is like A, C, or G. I might be wrong. Someone can correct me in the chat or something. If you can find Cas12 PAM sequence, I'm pretty sure that's what it is. It's like TTT, T for table, and then V. So that would probably work really well for organisms that are AT rich.
Right. And so it's Cas9 is beneficial for clammy because clammy is GC rich, so we will be able to find a lot of NGG You can
Hopefully, don't jinx
hopefully.
yourself, but
Yeah.
yes. I hate saying that, but I always say it anyway. I don't know if I can jinx
Okay.
myself. I don't know if that's a thing, but yes.
Okay.
Alvaro, do you remember what the percentage? Isn't it like 70 something percent of
Yeah,
the
something like
genome?
60 or 70, something like that. Yeah. But
Yeah,
it's
it's
an
like a
tissue.
large percentage of the genome is just G's and C's. So, all right.
Okay.
So, yeah, so this is basically, and this picture I pulled off of IDT, Integrated DNA Technologies website, which I've pulled so many figures from their website. They also have like really nice explanations if people want to just go read and look at what kind of systems they have. You don't even have to look at the prices or what they offer. There's just even their explanations honestly helped me quite a bit when I started working on all this stuff. The stuff that we were buying, the Cas9 protein and stuff, we bought from IDT anyway. So
Mm.
it just worked out
Yeah.
really nicely for me.
Yeah,
So
they're really
anyway,
nice figures. Yes.
yeah. All right. So this is how they adapted the CRISPR system so that you can customize it to targeting whatever you want. So what happens once your Cas9 comes in? Okay, you've designed your guide RNA so that part of it, this green part, is hybridizing to this the dark blue strand. You have your PAM sequence here. That's what this dark pink is. And let's say Cas9 comes in, successfully cleaves DNA. It creates a double strand break. At that point, this is, I mean, ideally you will get like a clean double strand break. There are two ways this will be repaired, right? And at this point for repair, you're not depending on this Cas9 system anymore. The cell's own DNA repair machinery should come in and try to be like, oh my God, our DNA is broken. We need to fix this. So it'll come in. One of the ways that this repair happens is called non-homologous end joining, or NHEJ, where it's kind of arbitrary the way that it gets fixed. So you might end up with an indel, which is an insertion and deletion, or you might end up with only an insertion, only a deletion, basically some kind of mutation. Now, this works a lot of times for, when people are trying to disrupt genes and you just want to see what the function is of that gene, or I guess the protein encoded by that gene. This is perfectly fine. But if you want more precise repairing, that is, you want homology directed repair, then you include this other DNA sequence, which can be double or single stranded, that has some specific sequence. But the key here is that the ends of your donor, it's called a donor sequence. The ends of your donor sequence, you can see this like the steel light blue color, have to be the same as the flanking sequences of where your cut was, right? And if you just Google or go look up YouTube for DNA repair, double strand break repair mechanisms, they'll explain very nicely how it is. I mean, it is pretty much necessary for you to have this part and this part match of your donor, and both sides of your double strand break in your DNA. So if ideally what will happen is the cell's repair machinery, if there's one of these donors floating around, it'll grab it, it'll use this as a template basically, and repair that break by using the template as a template, which means whatever is in between those light blue areas, which is green here, will get incorporated in your DNA sequence. So as you can imagine, in a cell, it is much more likely that you will get this kind of repair, DNA repair, than homology directed repair. Like there are certain stages of the cell cycle where homology directed repair will occur. And that is also not a guarantee. Like it's just more likely to occur during that stage of a cell cycle. I think it's the S stage, which is when DNA replication occurs, and also the G2 stage, because it's like, isn't it mitotic phase, G1, S phase, and G2? I
I don't
cannot
know.
believe
We
that I
are
remember
we are
this.
biochemists, so we don't
That's fine.
learn a
That's
lot
fine.
about
It's
cell
okay.
cycle.
It's okay. It's okay. It's always good to know, because as people will discover in research, there's like,
Yeah.
you're expected to know all
But this is
parts of.
so you're saying that this is cell cycle dependent, or is it like it doesn't occur, it occurs at specific point of cell division, like
Yeah,
these two
yeah. So
paths?
HDR, yes, it is, at least it is much more likely to occur in the S phase when DNA replication occurs in the cell, or in one
Mm,
of the growth phases, whereas
okay.
NHEJ kind of repair can happen anytime
Oh, I
in the
see.
cell, which is also like, you can imagine, I mean, it takes a lot more work to like find a donor floating around somewhere after this break has occurred. And the DNA machinery wants to get to work, it wants to fix this break in the DNA. So
Makes sense.
it's fine with just grabbing any random pieces of DNA lying around, or just the fixing in NHEJ can just be arbitrary. So you'll end up with this kind of repair a lot more often than
Yeah,
this.
yeah, that's why this is difficult. The HDR is more difficult to
Yeah,
achieve, right?
yeah, yeah. So anyway, so this is basically, yeah, this is, I guess I'm still continuing with CRISPR. So I'll talk about one more thing, and I'm not sure how much you guys have studied about this, but when you think of, if you want to express like a foreign DNA sequence, like a foreign gene, or I guess even an endogenous gene, something that's already exists in the cell, you want to express it in your cell, you people usually will think of a plasmid, right? Or like a vector, where you have like, you'll have a promoter, you'll have your gene sequence, you'll have terminator, you'll have some other parts of the plasmid, but it's basically like something like this that you see here. In this example, like you can see, they have a sequence for Cas9, they have a sequence for guide RNA. Each of these, like Cas9 will have its own promoter terminator, guide RNA will have its own promoter terminator. And you think of it as like, I mean, they use the word transfection, but we are more used to, I think that's more for mammalian cells. For like plants, we generally say transformation instead of transfection. So you'd introduce the plasmid into the cell, and then once it's in the nucleus, it'll get transcribed. Then the mRNA will come into the cytoplasm, and then it'll get translated, and you'll get your protein. Okay, I know that looks very simple, and this figure looks great, but it is a huge headache. There are lots of challenges to getting this to work, but that's basically what it is. So I'll explain. So ribonuclear proteins, or RNPs, the advantage is, you make your guide RNA outside. So you're not depending on like a plasmid or anything. And then you take your Cas9, you know, you I mean, I made this complex basically, but you order these two parts from IDT or some other company, and then under the right conditions, they will form a complex. It's basically just putting two things in a tube. It's not that complicated. Um, Once you have the complex, you deliver it to cells. It can still be the same thing. Like you basically do like an electroporation, and then everything. You hope everything else will go according to plan, which is the the DNA getting transformed into RNA. I'm sorry, what am I talking about? Wait, you already have your complex. So this complex will go scanning the genome in the nucleus and do the cutting. Hopefully the guide RNA finds its match and it'll cut. So you can imagine, if you go the plasmid route, which is the conventional method actually in many kinds of organisms, you have to first construct that plasmid. You design it, you construct it. It can be labor intensive, it can be time intensive, right? You have to make all the parts, put them together, you have to sequence it to make sure there are no errors anywhere. And the plasmids can get pretty big, because your Cas9 gene, I think, is like 4 kb or something like that. Your guide RNA can also be pretty big, and a plasmid typically will also have something that like an antibiotic resistance gene that will make sure you can select for cells that have taken up your plasmid. Because some cells will take this plasmid up, another cell will not. But you want to finally see only these cells after you do your electroporation. The way to do that is, you put your cells on an antibiotic, and since this plasmid will have a gene that can fight the antibiotic, the cells that have taken up this plasmid and start expressing that gene those are the only ones that you see. But don't confuse yourself too much with that if it's complicated. Anyway, so plasmid design or vector, sometimes people use those two terms interchangeably. Designing, constructing it can take time. It can take a lot of effort. Transformation efficiency is a problem. Just getting the plasmid into the cell itself can be quite a headache, because lots of times cells just don't want to take up foreign DNA, because it's kind of like they see something as trying to invade their body, so they don't want it. Expression also can be an issue, because remember, if you're trying to express these parts like Cas9 and guide RNA in your cell, they will need their own kind of like a good promoter that can drive that gene. Even if what is a good promoter, like the promoter might be good for driving that gene in a few different organisms, but in your organism maybe it won't work so well. So you can run into issues like that. Another thing is off-target effects. So when you have the plasmid in your cell, and then it's called maintaining cells with that plasmid, you keep your cells on the antibiotic to make sure that they continue to keep this plasmid. So you're keeping pressure on them to keep that plasmid. It'll keep expressing Cas9 and this guide RNA, and eventually the guide RNA will start making mistakes. I'm sure even from the get-go, they will make mistakes, but the more you keep producing it, chances of the guide RNA making mistakes, it'll go somewhere else and bind instead of the target DNA, and you end up Cas9 cutting all kinds of random places. So those are called off-target effects. And lots of times Cas9 can also be toxic to cells. So for all these reasons, plasmid-based genome editing can be quite complicated, although it is, a fairly conventional method of expressing foreign genes in cells. I mean, plasmids themselves. Here, of course, we're talking about specifically using plasmids for genome editing. Whereas RNPs, the advantage is you've already done the work of all this expressing the Cas9 and the guide RNA and all of that. You've already done it outside. You just deliver the complex to the cells, which is, if you're using electroporation, which is what we use in CLAMI, it'll basically create holes in the cells, and then the complex can come in if the cell is willing to take it up, and that's it. The complex will start scanning the genome for wherever, for a target match, and once it finds it, the Cas9 will immediately clip it. Eventually, within a few hours, actually, the Cas9 gets degraded basically in the cell. It's not like it gets to live there forever, because if it did, think about it, like we would end up with the same problem as we can with plasmids, which is having off-target effects. We don't want that. So you definitely take care of the problem of off-target effects and Cas9 being toxic to cells, to clammy cells specifically, by using an RNP method. Now, as you can imagine, life is not that simple. So this probably, this is more expensive than trying to do a plasmid-based system, even though it is far more efficient. Now, I mean, with time, like things, when they first started this itself, from then prices started coming down as more people started using RNPs. So that will hopefully not be like a limiting factor. The cost being a limiting factor will not be a thing for that long. But yeah, the one downside to this whole system is that it can be quite expensive to buy these parts. Unless, I don't know if people start making these parts in-house. That itself, I feel like we'll go back to the problem of labor and time intensive.
Do you know how expensive if it is like to order the guide RNA and Cas9 from, is it from NEB, right? They have,
Well, so in that lab in Lincoln, we bought it from IDT,
oh,
from Integrated DNA Technologies. Um,
is
I
it
can
like,
tell you, yeah, I mean, it's pretty expensive to buy like one aliquot of Cas9. I think it used to be like $500 or something, but that tube would last you for a few electroporations. I cannot give you numbers off the top of my head. So yeah, it can still be like, maybe like one big CRISPR experiment that I would do would probably be a few $100s or something, which is why, I had to make sure like everything was prepared. Like, when you grow your cells, um, this is something we didn't do much in grad school, but the day I was going to do like a CRISPR experiment, first thing I would have to do come in the morning is look at the cells under the microscope and make sure that they don't look stressed, which means, like the cells, if they're in clumps together, that means they're probably going through some stress. And if I would see that the cells are stressed, that's it. You have to abort the experiment and restart growing cells. Because if they are not happy, then going through this whole experiment would be, obviously it's a waste of money.
okay, okay, yeah.
So that was one example.
You need to use happy cells because
Yeah.
expensive
Yeah.
Cas9 and guide RNA.
Yeah, exactly. So, all right. So, anyway, so these are some of the challenges of using plasmids versus ribonucleoproteins. And of course, I mean, the challenge of using RNPs is also there where, it can be expensive and even handling it, you have to be careful and not just, like, for example, I had to keep things on ice here and there. If you're like chit chatting while you're doing the experiment and you casually keep holding the tube in your hand, yeah, you'll probably end up with like inefficient Cas9 and guide RNA that you got from the company.
Okay, okay.
So yeah. So uh, the goals, well, before I talk about our goals, I'll say, so before we had done this experiment, people had, successfully used CRISPR-Cas9 for genome editing um, in clammy. It's not that we pioneered it or something. People had generated knockout mutants. So let me go back to this example here. So people had basically done something like this, where this whole green portion in the donor would be, for example, an antibiotic resistance gene. So that's so convenient, right? You have your target gene, you use Cas9 to like create this break. And if the cell uses this donor DNA successfully for as a template, you would have disrupted your gene already because there'll be this like huge piece of DNA in between. But that piece of DNA in between is what? It can be like an antibiotic resistance gene. So you've actually taken care of two problems at the same time, where you've disrupted your gene of interest, but you've also put in a way that you will be able to identify the cells that have your gene of interest disrupted because they will survive on the antibiotic. Because this gene here, will, whatever, it'll get expressed, it'll make some protein will help fight the antibiotic, basically. So people had made like knockout mutants. That was one of the main things at that point, that knockout mutants had been generated using CRISPR-Cas9. What we wanted to try, or I will also preface all of this by saying part of this project was, of course, it had already started by the time I got to that lab. And of course, this is like the brainchild of two professors in UNL and in Lincoln, Nebraska. It was Dr. Surudi, who was my boss, and it was Dr. Weeks Alfredo. Both
Yeah,
of them. This whole project was their brainchild, basically.
yeah.
So they wanted to implement like precise editing of genes, where basically you only want to change like a couple of bases in your target DNA. So for this, we achieved this by co-targeting a gene for selection and a gene of interest. So two of our genes of interest, one was called FDSY, which is involved in chlorophyll synthesis, and WDTC1, which is a putative anti-adipogenic factor. So when you see adipogenic, think of adipocytes, which are fat cells in our body, right? So adipocytes, or you can think of fatty acids or lipids basically in plant cells. So it is an anti-lipid, anti-fat factor, they think, based on studies. So basically it means that when this gene is expression is low, there can be more fat accumulated. Normally when the gene is normal expression or high expression, it means it's anti, like it does not produce as many fats. And when you think of something like lipids and fats and stuff in plant cells or bacterial cells, you can immediately think, why do people care about that? They want to use it as a source of biofuels, right? If you can get the cells to express or make more fats, you can harvest that fat in a way so that you can use it as fuel, biofuels basically. So, and I'll explain why we were interested in the FTSY gene. That was basically because you'll see, I think I have it on the next slide. It's because when the gene is disrupted, it's very easy to identify the colonies on plates, because the protein coming from this gene is required for insertion of the light harvesting complex into the thylakoid membrane. So when this gene is disrupted, this whole uh, process does not take place properly, which means you end up with like this pale green phenotype. So that was the reason that we chose that gene, because they'll have low chlorophyll content. You'll end up with a pale green cell, and it's just easy to identify once you do your transformation. So for selection, that was our gene of interest. Now, remember, we talked about selection is also important, right? Because what's the point if you simply throw all this other stuff, your CRISPR system, into cells, and then if you, after you do the recovery and whatever, you plate the cells, everything will just manage to grow on the plates. You need a way to kill off a bunch of the cells. That is called selection. So for that, we use a promomycin resistance gene. Promomycin is just an antibiotic. Don't worry about that too much. So what all did we include when we did this electroporation? One, we had this whole CRISPR-Cas9 complex. Okay, remember this light blue blob is your Cas9, and this green and orange or brown part, whatever this is your guide RNA. This bold part of the green, we have designed it so that it will target this gene. That's your Cas9 RNP complex. Then we did experiments where we added a donor DNA and some in which we didn't. And we created this donor DNA so that it would have like very specific changes. If it is used as a template for repair once Cas9 does its thing, then it will replace just a few bases, and I will go over that in the next slide. So either we included the donor or we didn't. And the third important part we included in the electroporation is just a promomycin resistance gene cassette. So that would have its own promoter, a terminator, and then in between the gene itself. So I'm not sure how familiar you guys are with the concept of antibiotics and stuff, but that's basically what it is. It's like if a cell has this gene and it's able to express it, that protein will help fight any antibiotics on the plate in the medium where the cell is trying to grow. If the cell doesn't have this gene, it won't be able to survive. The antibiotic will kill it. That's basically what it is. So once we did this, okay, so I'm going to go over a little bit of, this design process. So the target site that we chose in this FTSY gene was in the 4th exon of the gene. You can see all these green bars are basically exons in the gene. And the CRRNA, which is the part of the guide RNA that recognizes your target sequence. So this sequence, you can see it would bind the strand that you see here that is in the genome. And there is a PAM sequence right here. And remember, PAM sequence for Cas9 is NGG. So you can imagine on the other DNA strand, the sequence would have been GGG, which satisfies our requirement, right? It's NGG. N can be anything. So, so that's the CRISPR RNA, which is part of the guide RNA that helps guide the Cas9, the RNP, to our target site. And our target site was right here where this triangle is. And then remember I said in some experiments we included the donor and some we didn't. The donor had these specific changes where this CCC, it's a little bit confusing here because it overlapped with the PAM sequence in this example, but this CCC would be changed to a TAG, which is a stop codon. Okay, so it will introduce, if it uses this donor successfully for, what's it called? For repairing the double strand break, it will change the CCC to a TAG, which is a stop codon, and it'll also change these two bases to different, so TC to AA. And in the process, when it changes the triple C to a TAG, it'll also create a restriction enzyme recognition site for NHE1. There are reasons for this. Now, premature stop codon, you can imagine, why do we want that? If you successfully introduce a premature stop codon in your first exon, when you can see there is so much more to the gene, you're going to end up with a very truncated protein once that is actually expressed, which will achieve our goal, right? You've changed things very precisely, and you've most likely disrupted the protein. You'll end up with such a small protein when normally the wild type protein is going to be much bigger. So that's what the premature stop codon would be useful for. Creation of the NHE1 site is useful for screening, and I'll go over that. I think it's on the next slide. So when we did this, we found that about 1.5 to 2% of surviving colonies, which are of course resistant to promomycin, they had a disrupted gene, which means there were insertions or deletions. DNA cleavage was in the expected target site. So at least our Cas9, the whole complex was doing its job of finding the target site, and then Cas9 like actually cleaving the DNA. But the thing was, none of the edits were using our donor. So remember, I went over two types of repair mechanism results, right? There is NHEJ, which is just arbitrary. It'll somehow just fix the double strand break, or it'll use a donor and it'll nicely use a template and put whatever changes you want. That did not happen. Only NHEJ kind of edits happened. So it didn't use the donor, even in cases when we actually put the donor in our like transformation mix. So now I don't know how I'm doing on time. Like I said, I can talk quite a bit. It's already been an hour.
Yeah, it's in an hour, so yeah, we can, just go ahead.
All right. So ProTox, I'm going to give a brief introduction to what ProTox is, and I'll explain how it comes into play. So ProTox is an enzyme that plays an important role in the biosynthesis of heme and chlorophyll. Basically, what you need to know is that in the presence of a specific herbicide, if ProTox function is curtailed, what happens is, it's supposed to convert this substrate, which is protoporphyrinogen 9 to protoporphyrin 9. If the herbicide doesn't allow this enzyme to do its job, this thing will go get transported to the cytoplasm. It gets oxidized, it gets accumulated there, and basically it will lead to cell death. So that's what ProTox does. So if the herbicide is there and you have a wild type ProTox enzyme, just understand that it will kill the cells. So, but what people have found is that with a single base pair mutation, valine at position 389, if it's converted to methionine, then it leads to herbicide resistance. But the mutation doesn't affect the function of the enzyme. It can still do its job. It's just that it becomes resistant to the herbicide. So you can imagine this is a very specific change. So what we did was we designed our CRISPR RNA so that it would make us, wait, don't I have that? Yeah, okay. So that it would change that G to an A. And then this was just like a mutation that wouldn't have any effect. But we designed the donor so that it would create these two changes in the wild type gene. Because remember, if the cell uses the donor as template for repair, then it will be able to be resistant to that herbicide, and it can still do its job, like Protox can still do its job. That's basically what it is. So I think this is all the same thing, basically. Yeah, so that C to T mutation was a functionally silent mutation. I won't go into it too much, but that was basically because we were trying to see if it would keep using the template. It might incorporate this change, so your cells will become resistant, but we wanted to see if it'll keep going for a few bases after, because, it's not like the donor will stay there forever. Sometimes it'll just fall off, and the repair will not be according to the donor 100%. Anyway, so about 2/3 of examined colonies we found were edited by HDR using the donor as a template. Now, so we went back to FTSY, and this time we had two Cas9 like RNP complexes. So one, you target Protox, so you have to have that donor that will make those two base changes. And then you have FTSY, which will also make those other base changes. So if you remember with FTSY, what we wanted was the premature stop codon, and we wanted, which would also create this recognition site. Now, in these PCR gel pictures that I'm showing you, G1 was our wild type. Okay, this 3261 and 31 were other what's it called, they were transformants. 31 you can see clearly. Another thing to think about is if it uses your donor exactly to create changes, then you won't be able to tell by PCR, because it's just a base to base like change, right? You've not changed the length of your DNA, your gene of interest DNA sequence. So when you do PCR for these two, you can see the size is exactly the same as the wild type. 3.1 is an example where it didn't use HDR. The repair happened so that it went and put like some other DNA, probably part of the donor DNA itself, it went and stuck in there. So, but the neat thing about this method is, since we introduced a restriction enzyme site, you can take the DNA from these PCRs and add the restriction enzyme to it and run it on a gel. If it used the donor and incorporated these changes in here, what will happen? Your DNA will get cleaved nicely. So once we looked at this, that, okay, so it's not just wild type DNA, it actually incorporated your own gene changes. And yeah, G1 is just wild type, so you can see it's not the same size as G1. So that is the utility of adding like a restriction enzyme site when you do this sort of thing, because screening becomes easier. I mean, this is just initial screening. This is just to screen out ones that didn't get edited, maybe. But even after this, we would send things for sequencing, because especially when you're trying to publish, you have to make sure that it actually is the changes that you are saying were there actually got incorporated. Even this much evidence is not enough. So anyway, so these are a whole bunch of different mutants. You can see like these are all the NHEJ repairs that we talked about, right? All kinds of changes got incorporated here. And yeah, the first three ones you can see didn't get anything incorporated in the double strand break site, but the changes that we wanted from our donor came about, which means those were all HDR, what do you call them? HDR edits. So of 223 herbicide-resistant colonies, 9 had this pale green phenotype, so they were all mutants, which is 4%. I know that doesn't sound like a lot, but transforming clammy, getting edits, that to specific site-directed edits can be a pain. So this is not a huge number of colonies to screen, honestly, even though that number seems really low. And of the 9 that had edits, only 3 had precise editing by HDR. So 1.3%. So when we used the promomycin gene and we didn't target Protox, we didn't get any HDR edits. When I say promomycin gene, when we just put the gene cassette in there. And then But then when we also target protox, we got about 1% that clearly, remember, if you targeted protox and your gene of interest, definitely the protox also would have had like that one specific base change, because otherwise the cell would have just died. So that means HDR edit took place in protox and your gene of interest. Okay, so WDTC1 is just another example. I'll go over it quickly. So they found that, in flies and mammals, loss of function meant increase in adipocytes and fat accumulation. In Arabidopsis, mutants in a homologous gene, they saw that the seeds had an increased fatty acid content and greater weight. Again, it comes down to the same thing where they want to see if they can use these organisms as a source of biofuels, basically. Which is why usually they're interested in lipid levels. So again, we design an RNP that we target this WTTC1 gene. You have a donor, and then we tried it with promomycin resistance cassette. Excuse me. So of 96 antibiotic resistant colonies, 2 had alterations, 2%, not terrible. But then none of them had HDR edits, even though we included the donor in the transformation. But then, lo and behold, you can guess the moment we also target protox with the donor, that is, we're looking for a specific change in protox, which will allow the cell to survive, of whatever, close to 400 herbicide-resistant colonies. 9 had alterations, 2%, not terrible. Of those 9, 3 had precise editing, which is only about 0.8%. So yet again, what we saw was if you just throw in that promomycin resistance gene cassette and you give the cell donor, the Cas9, or sorry, the repair mechanism is not interested in your donor. But if you try to target protox along with your gene of interest, both with like an RNP, a Cas9 complex, you will end up with some that will use your donor as a template for double-strand break repair of your DNA. Now, okay, so I've come to my summary. Sorry.
Oh yeah, go ahead. Okay, so I'll have a question about that one, but
sure. So I hope you're seeing the pattern here, and you can kind of guess why the cell is willing to use your donor for your gene of interest, like this FTSY WTTC1. They're using your donor because you have already selected for cells that already are using the same mechanism to edit the gene, which is going to allow the cell to survive, right? I hope that's not too confusing. So basically, the likelihood of your gene of interest getting specifically edited by HDR goes up if you have already forced the cell that you're looking at to have already used HDR as a way to like fix something in the cell. And there is a clear bias here because our population, if you're already looking at it and it's already surviving on the herbicide, that means it used this protox and this donor, this whole thing, to perform HDR. So it's likely what's happening in the cell is it's like, oh, we've already performed HDR here. Let's go try to perform HDR here also and use the donor that they provided for gene of interest.
But
whereas,
you do it
sorry,
all the time, right? So you introduce 2 RNPs all together during transformation?
Yeah. But you have to make them separately. So you make like one complex, you make another complex, and then you add the donors. But then everything goes into the tube with your Chlamydomonas cells, and you put it in the cuvette and do electroporation.
Okay.
But you have to design, your one guide has to target protox, one guide has to target WTTC1 or whatever your gene of interest. And your donors also should be so that they have those whatever specific base changes you want in your gene of interest. So the donors will be different. I should probably say like donor for protox and donor for gene of interest. So to summarize, we were able to use CRISPR-Cas9 RNPs to correctly target. So finding the site is important, right? That's not a given. So able to correctly target and cleave DNA sequences of interest. So when we used this antibiotic resistance gene cassette for selecting transformants, we did not see any homology directed repair edits. Everything was that NHEJ where your gene is somehow disrupted. So about 2% of transformants have disrupted targets. Whereas if you're co-targeting two genes, one for selection of transformants, so in our case it was Protox, and the other is being your gene of interest, and you're using the RNPs with donor templates, you can end up with precise homology directed repair edits in both those genes. You will still get ones that have that are like NHEJ edited, which means they have some sort of disruption in the gene, but you will get 1%, about 1%, that are precise and edited without a scar, basically, because it doesn't leave behind anything in right, because the Cas9, that whole RNP eventually will get degraded, and yeah, and your gene of interest is, that's why we call it precise scarless editing. You've not introduced anything into your own gene other than the specific changes that you wanted. So that was the whole thing. And like I said, so I made a QR code for the paper. If you guys want to read it, feel free to. Oh, I didn't include my email, but it's just my first name dot my last name at Gmail. You can get it from Alfredo. Feel free to email me, or you can ask him questions. He can come and ask me questions. But what I wanted to say was, this project started before I had gone to Lincoln. So of course, credit to all the other people who are on the paper. And yeah, it was the brainchild of two professors, one of whom was very good friends with our grad school professor, with Dr. Spalding. And both of them, yeah, they are the ones who came up with a lot of the ideas here. So
Right. Yeah. So
yeah.
this is the paper, right? Okay.
Yeah.
Is there
Okay,
anything?
I was really worried that I would not be able to talk for 45 minutes.
No,
Apparently, I've been talking an
you're
hour
good.
and a half,
It's
or
really
hour and
nice. And
15
then
minutes.
I recorded this because some of our colleague the lecturer or the professors here, they're interested in this lecture, but they cannot attend because they have classes. So I recorded it and I'll share it with them.
Sure.
Okay, now question times, right? If you guys question, mungkin dari mahasiswa saya mungkin yang ngambil kelas ini ada pertanyaan, silakan. Almer, mungkin Rahel, yang lain, Stephen, ada pertanyaan? Bisa dalam bahasa Indonesia, nanti saya sampaikan dalam bahasa Inggris. Pertanyaan yang standar juga nggak apa-apa sih, kalau ada pertanyaan. Silakan. Dari mahasiswa dulu. Saya punya banyak pertanyaan sih, cuman dari mahasiswa dulu. Ada pertanyaan atau dari Bapak Ibu lain, mungkin dari luar yang ingin bertanya. Saya paham mungkin ini suatu konsep yang cukup baru ya. Mungkin di Indonesia belum banyak yang melakukan penelitian di bidang ini, tapi kalau ada pertanyaan walaupun sangat basic, silakan bisa ditanyakan. Okay, so my question is there any specific length of the donor DNA? Did you optimize it, like affecting the HDR repair mechanisms?
Yeah, if I remember correctly, it's only about roughly 20 base pairs or so. I might be wrong, I'm sorry, but I think we also didn't have to optimize that much because other people have used, I mean, they haven't used donors for specific edits, but they have used donors before. So I think we went off based off of that. Hold on.
But and also there's like any like concentrations limit, the limit concentration required for this, or if you put bunch of HDR donor DNA, you will induce HDR more, or is it more dependent on the, if you targeted two specific
Both.
sites all
Yeah, I
together.
think, yeah, that's probably more important because obviously whether or not it uses your donor is more important. Like the limiting factor is usually not whether or not there is donor, right? Like we saw when you have like promomycin resistance versus, I'm sorry, like introducing antibiotic resistance gene versus targeting another gene, that protox gene for what's it called? Sorry, for selection.
Mhm.
The more important thing is that you make sure the cell is actually doing that HDR repair
Oh,
rather than how much donor there is. Because even when you include donor, but you just give it that promomycin resistance gene cassette, it's refusing to use the donor. So it's
mm,
more important that you catch it, which is also why we had to, I mean, of course, like, trying to get cells that are completely synchronous can be difficult. Um, but yeah, the idea was you use kind of like young cells, which are likely to be going through like lots of replication, right? That's
mhm,
why that was another factor actually, because like I said, HDR is far more likely to occur in like that one growth phase, which is S, not
mm.
one, actually two growth phases. The one of the growth phases, G2, and the S stage of the cell cycle. So for that, you want to make sure that the cells are actually like active replicating stage, because normally there is like a growth curve, right? So by the time, if it starts growing, takes off, and then levels off, and you catch the cells at that stage, it's likely they're already, some of them will be dying. It's like they're going through
Mm.
senescence. They're too
Yeah.
old at that point, most of them. So it's better to catch them while they're actively growing, which is when it goes through like all that DNA replication and all of that.
I see, I see.
So
So it's it is important to have like co-targeting of genes to
yeah,
induce HDR,
yeah,
and
yeah,
then cell cycle is also important. Like when you
yes,
introduce this is very
yeah.
critical for
Yeah,
achieving the
which,
goal.
I would imagine that's probably true for even if you're just using a plasmid to overexpress some protein, even when it doesn't have anything to do with CRISPR, it's probably always better to use cells that are going through like an active growth stage rather than once they've reached stationary phase. So I
So
think
in
that is,
chlamydomonas, well, when I did transformation back then, I like, yeah, we have like certain protocol, right? Like you have
yeah,
to keep
yeah,
them for 1 or 2 days, right? Something like that
yeah,
from the stomach
yeah,
culture, and then you have to remove them. So we just used like by eye, like the color, like, okay, this is
yeah,
green, and then
yeah, yeah.
so is that the right thing, or like what's what's
I
the
think,
right
I mean,
thing?
honestly, I have to say, I felt like an idiot sometimes when I used to do these CRISPR experiments in this lab because I was paying so much more attention to these things than in grad school, where, like you said, we just kind of eye it and go by that, whatever the protocol was. But yeah, it's
That's
good.
why it
It's good
failed.
to, I know, it's good to check OD. I
Oh,
mean,
D.
you probably will have like some sort of established something. I mean, you can establish, if you're working with any organism, you can establish some sort of growth curve, do it a couple of times, grow them and keep taking like regular intervals what the OD is. Then you'll figure out like, at what stage they are not yet taken off and then they start going up, like
Yeah.
the OD starts going up and then it levels off. So at that point, you'll know, okay, roughly this OD this to this is when they are still in like replicating stage, number
Mm-hmm.
one. Other thing, like I said, I would look at the cells in the microscope, under the microscope, and see like, are they looking clumped? If they are clumped, then it's for this particular experiment, it's not worth going forward with, like, because there's like you're spending a bunch of money. But even with
Yeah.
plasmids and stuff, I would recommend. Again, like it all depends. Like it's a trade-off. If you want lots of transformants, or if you just want one, and you're like, even if the cells are like almost in stationary phase, you will end up with uh, a couple of transformants, and that's all you need. You just want one mutant. That's different. If you're
Mhm.
just trying to overexpress something,
And
whereas if this is like a methods study, right, where we needed lots of transformants, and then you have to like screen them and all that. So it was important definitely to like make sure we had like optimal conditions. So that was one of the things. So
So do you
yeah,
think
we did. Do you remember checking? Did we check OD when
I think I
in?
did. I checked the OD, like I try to use the same OD, but some I don't know, or I just use like sometimes I'm like too lazy, so I just like use it by
I know. Yeah, lazy, and it's not just OD. I would also count cells in the
oh
microscope
wow. Oh, I
to
see.
because that is considered a more like reliable way of measuring like cell concentration, number of cells. So
Mm. Yeah,
yeah,
yeah, yeah, yeah.
but
So growth curve is important, and happy cells under the microscope is crucial for
yeah,
transformation. So
yeah.
the cell wall, do you think it is important? Like the clammy cells with cell wall and wall cells, or they are also critical?
Yeah, so it's a great point. Of course, I think there are a couple of parts of the paper that I completely skipped we didn't talk about here. But that was another experiment I did where, you know, so basically what happens is there are two mating types in clammy. Alfred already knows this, but I'm saying
Yeah.
this for everyone else basically. But there are two mating types in clammy. When you they're called mating type plus and mating type minus. When you make them mate, so you mix them first, you have to, in order to, I mean, it's called gametogenesis, where if you deprive the cells of nitrogen, they will become gametes. Basically, it's not like they are making another type of cell or something. The cells themselves will become vegetative cells, become gametes. So they'll be either plus or minus. If you mix them together at that point, they make proteins that will basically digest their cell wall so that they can mate. So that protein is called autolysin. You can imagine lysis means breaking, right? So autolysin. And so I actually made, I mean, I didn't make the cells, made the autolysin, which means I would grow up like two wild types. Alfredo, you will know CC620 and 621,
Okay, okay, yeah.
those two strains. You basically grow the cells, then you transfer them to nitrogen-free media, so they'll
Mm-hmm.
become gametes. You
Yeah.
mix them, and the protocol is there in my paper maybe, but I
Yeah.
also had to try, like, you make them mate for different periods of time.
Mm-hmm.
And I think I eventually settled on 4 hours. So at that point you halt the mating, which means you just spin down all the cells, pellet the cells, and at that point, like, the supernatant will have that autolysin.
Oh, I see. And then
So
you use it.
I would save that supernatant in the freezer. And whenever I would be doing this CRISPR experiment, it used to be such a long day, but I had to do this other step of, like, your cells that you grow in which you're going to do electroporation, you treat them with that autolysin, and that autolysin will digest the cell wall, it is an impediment to, like, it is a barrier for electroporation, right? Because you have cell wall, then you have cell membrane, and you're trying to get all this stuff in there, and sure, you're sort of like applying this large voltage of electricity and you want holes in everything, but you have so many problems. Like, you have the cell wall, you need a hole in the cell wall, you need a hole in the plasma membrane, you need the cell has to be willing to take up this foreign DNA. So you would have removed one of the barriers. Basically, it will digest the cell wall, and that actually showed like huge improvement in our numbers when we
I
used
should I
autolysis.
should try that for the clammy transformation,
Yeah,
right?
definitely ask me if you want. I have like detailed notes somewhere, and you don't have to like go through
Perfect.
all the trouble. I can help you with some of that stuff.
Perfect. We can try
Yeah,
that. Okay.
yeah.
Yeah. So, guys, do you have questions already? Or if there is no questions, any questions maybe from the audience? Yeah, there is one question from Gogo. Gogo's, go ahead.
Yeah. Hello. Can you hear my voice
Yeah,
clearly?
I can hear you.
Okay, so I'm curious about your cell culture technique. How do you prevent the cell clumping during your experiments? Do you just regrow it, or did you add any reagents or modifying the growth conditions of the cell?
So typically one of the things that apparently is the common way that clammy cells get stressed is if if you're in your glassware, in your flasks, there is if there's like detergent, because it's always going to happen when you wash the flasks, there'll be some amount of like soap, like detergent left behind. So to prevent that, what we would do sometimes is, this is a common thing. This is not specific to clammy. You take your glassware, fill it up with water up to like some height, cover it up, and you autoclave it. And then when it comes out, you just dump out all the water, and then you put your media in there, and then close the flask and autoclave it again in order to like make your media sterile. And once it comes out of the autoclave, you let it cool, you add your cells. So that part of growing your cells is the same. It's just that instead of washing your flask and immediately adding, your media and autoclaving that and then adding your cells, you put it through like this autoclave cycle where you just autoclave it with some water, some distilled deionized water, DDH2O, right? You just autoclave it with that, and that's supposed to help like peel off any detergent on the walls. But
So
I don't know. If you have like nowadays, there's also like all these detergents that are specifically made for lab glassware that will not leave behind any residue. So if you have access to that, great. But sometimes I used to just do this as a precaution anyway, because I would not want to come in on the morning and see that, oh, I have to abort this whole experiment. I wanted to do it if I had planned for it. So that's
that's a
one thing.
great strategy. Sorry, can you repeat that again? I get like a little missed on that.
Sure.
So
Yeah.
you autoclave your glassware along with the detergent at the same time?
No.
Or
So you have washed glassware, right? That you have washed with detergent.
okay, yeah.
And then normally
Yeah,
what would you do? You wash it, you leave the glassware to dry. And
right,
then once it's dry, you add media in there,
right,
autoclave that.
right.
And when it comes out, you cool it, and then you add your cells whenever you're ready.
Right.
But this is if there is any detergent stuck inside of the glassware when you added the media, that can cause stress to the cells,
Oh,
apparently.
oh.
So what we do is before you add the media and all that stuff, you wash your thing. You don't even have to wait for it to dry at that point. You just add like water at least to the level that you expect to have media in there. And actually I would say like halfway through the flask, just add some DDH2O,
Yeah.
close it, and put it in the autoclave liquid cycle.
Oh, liquid cycle.
It's
So it's not like full autoclave mode. It's just to heat it up.
Yeah, basically. And
Can we?
then so it's almost like if you wanted to get sterile water, what would you do? You'll add some water to the flask and put it in the autoclave. When it comes out, basically your sterile water, but we don't care about the water. The idea is going through that autoclave cycle with the water apparently helps peel off any like residual detergent on the inside of the flask.
Oh, I see, I see.
Yeah.
I
So
think my students should have done that. Like once you rinse it, because we use like really strong detergent for that, and sometimes the cells get clumped.
yeah,
You have to
yeah. There
autoclave
are
it.
some Yeah, there are some detergents that are sold that are supposed to be like, they say they won't leave any residue on the
It's
glassware. I'm sure
expensive.
those are very expensive.
It's very
Yeah.
expensive. You know, I
Yeah.
checked that, and it's like the price in Indonesian money is about
Yeah.
uh 20 million rupiah. So it's about how is it like thousand $1,500,
Whereas
$1,500.
whereas what? Like regular detergent will be what,
Oh,
10,
it's like
$20?
it's like $2 or something, and you can
Yeah,
get it for
yeah,
like whole months.
yeah. So it's better
Oh,
to go through this extra autoclave cycle.
okay. Yeah, that's
Yeah.
good. Good tips. Thank you. Any,
Okay.
is there any more questions from students? You can ask anything, by the way, right? Soujanya,
Yeah, yeah,
or see
please. Yeah,
some
whatever you want to
experience
know if
working, or if you want to go for like grad school in the States and everything.
yeah.
Mm.
Yeah, if I mean, of course, you have a great resource in Alfredo himself there, but yeah, feel free to ask. You guys want to know if we were like playing the fool in the lab at Iowa State?
Yeah, that
Basically,
was like
I like to say this, we would cry laugh our way. Basically, it's like walk into the lab and you're like, why does Clammy hate me? Nothing is working.
yes. Oh yeah, I remember those times. Okay, any more question anyone from dari anak-anak ini TAB ada pertanyaan nggak? Yang kemarin udah baca paper CRISPR Cas9 ada pertanyaan?
Yeah.
Okay, if there is no questions, I will use this material for the exams for my students. So is it okay if I share it with them is it okay if I share it with them this
The recording?
yeah, this recorded
Sure.
presentations?
Sure, no problem.
Yeah. Oh, there is one question from Daniel. Okay,
Sure.
Daniel, go ahead.
Thank you for the chance. I'm really intrigued about designing sgRNAs. So what's the principles of choosing the proper sgRNA? Because like sometimes I see like people choose the PAM sequence downstream of the DNA, and sometimes people choose the PAM sequence upstream of the DNA. So which one is the proper one for designing a proper sgRNA?
Oh, I didn't know that people actually use like the downstream. I'm trying to go back to my presentation. You guys can still see my presentation, right?
Yes, we can see that.
As far as I know, we use ones that are downstream, at least for Cas9. Your PAM sequence is downstream. And typically, I think it's supposed to be 4 to 6 bases gap between your target, like, the cut site and the beginning of the PAM sequence. That's an interesting question. I don't know. Did you happen to see if like maybe it was for a different type of Cas nucleus, like Cas12 or something?
I believe the paper I saw was using Cas9.
Okay.
Yeah.
Huh. I should probably look that up. I'm sorry, I don't actually have an answer for you, but I can try to look it up and maybe I'll tell Alfredo so that I can try to find out.
Daniel,
And yeah, I mean, there are so many websites available nowadays, but definitely IDT will have some information on that because they help you go through this design process and then buy their products, obviously. But
Cool.
there's information there. Let me look it up and I'll communicate with Alfredo. Thanks for
Awesome.
that question, Daniel. Yeah.
Okay. Any other questions? All right, if there is no questions, thank you so much. Thank you so much, Soujanya, for your time and explaining to us about CRISPR and then the application in Chlamydomonas. And it's really nice to see you again.
Yeah, same.
And
After a long time. Yeah.
I don't know when. Yeah. So good luck with your postdoc and your research. And
Yeah. Same to you
maybe
guys
we
and all
will
the
invite you
students as
again
well.
sometime. Yeah.
Yeah, definitely. Yeah. Thanks to you guys and good luck with all your exams and everything. Yeah, thanks for giving me this opportunity.
Okay. I'll
All
stop
right.
the video and